we expect that we'll have a few more people show up, but I gather last night was a big night in Washington. Everybody was all over different events, including myself. So people may be running a little bit late. Anyway, I do appreciate you all being here so early in the morning, regardless if you were up late or not. It's still early, and I know that, so thank you. I'm Phyllis Greenberger, President and CEO of the Society for Women's Health Research, and I want to thank you all, and particularly the panel, for coming here and working with us on this, what we think is a very important issue, and I hope you do too. For those of you who don't know us, and I hope you do, or will after this session, we've been the leading voice in research into the biological differences between men and women. Actually, next year will be our 25th anniversary, hard to believe. We were founded in 1990 by Dr. Florence Hazeltine, who is sitting right there, also with a group of physicians, medical researchers, health advocates. We've been bringing national attention to the variety of diseases that affect women differently, disproportionately, or exclusively. And now, I'm happy to say, though we're not totally successful, but due to our efforts, women are now routinely included in most medical studies, and scientists are increasingly considering biological sex differences as a variable in their research. I'm sure all of you are aware that just in April, the NIH changed its policies to include female animals and cells in basic research. So today we're focusing on molecular diagnostics, which are tests that I'm sure most of you know this, that detect gene sequences and proteins to help diagnose and monitor disease, assess risk for disease, and determine what treatments will work right for what patient. And we are pleased to have an incredibly distinguished group of experts with us. Dr. Eric Green, who's the director of the National Human Genome Research Institute, the largest organization in the world dedicated solely to advancing human health through genomic research. Dr. Rita Colwell, a past SWHR board member and distinguished university professor, both at the University of Maryland and at Johns Hopkins University Bloomberg School of Health. She's a senior advisor and chair emeritus of Canon U.S. Life Sciences and president and chair of Cosmos ID, Inc. And Evan Jones, managing member of Javen Capital, LLC, a life sciences investment company. And to illustrate the importance of this technology and give you a taste of it, if you haven't been very involved, we have two demonstrations, both of which are, one is exclusively for women and the other has been found to be much more successful in women. So obviously, this is the reason that we picked these two in particular. Of course, I think you all know that there are many diagnostics out there now, and it's extremely exciting to see all the research and innovation. But we're going to begin with welcoming Dr. Eric Green, if you'll come up here. Thank you. Do you want to? You don't have to stand up here. You can roam if you like.
You can keep the microphone. They have me.
Okay, you're Wyatt. All right, then we better turn this on.
There we go.
Turn this on.
It's appropriate because I'm going to really sort of lay the landscape of what I think is going to be some exciting things you're going to hear from subsequent speakers. And the landscape I'm going to describe has really come about because of rapid enhanced knowledge about our blueprint, our genome, massive advances in new technologies that will allow us to study this blueprint in ways that previously were unapproachable, combined with the era of big data and stronger computational skills that allow us to mine information of biological relevance. Now, of course, the real foundation of this is the desire to understand the underpinnings of our blueprint, which, of course, is housed in the nucleus of all cells in structures called chromosomes. And it was the recognition in the late 1980s that our knowledge of DNA and early technologies for being able to study DNA put us at a point in history where it was imaginable that one could completely deduce the sequence of the 3 billion letters, the G's, A's, T's, and C's that make up the human blueprint. That, of course, led to the launching in 1990 of this audacious international effort called the Human Genome Project, which I participated in from day one, with this audacious goal of reading out the 3 billion G's, A's, T's, and C's that represented the human blueprint. This effort was biology's equivalent of the moonshot, if you will. It was remarkably successful, completed not in 15 years as originally imagined, but in fact was completed ahead of schedule, achieving all of its goals. Now, with that said, a lot has happened in the last 11 years, and much of the attention of the human genome's research efforts have been increasingly focused on a number of things that would put us on a trajectory to eventually apply this knowledge to improve the care of patients. And this has led to emergence of new fields such as genomic medicine, which is a major focus of the research that we support at the institute that I direct. I use the phrase genomic medicine as one to mean an emerging medical discipline that involves using genomic information about an individual as part of their clinical care and other implications of that clinical use. You'll hear other phrases like precision medicine, individualized medicine, personalized medicine. We tend to focus our attention on the genomic aspect of it, but these are all largely synonymous. Not treating patients generically, but treating them based on knowledge of their unique genomic makeup. Now, this is not going to happen overnight, and I think the other speakers will echo some of this. This is a journey of progress that's going to be required to really realize the potential of using genomic information in the care of patients. The starting line for this was very much the Human Genome Project, and the finish line, if you will, is going to be when we realize this on some reasonable scale. But there'll be many steps involved, and we don't even know what all those steps are at the present time. But in rapid fashion, let me just tell you some of the highlights of what has happened over the last 11 years in this journey en route to genomic medicine. And I really just want to tell you briefly about 5 major steps, collapsing really over a decade's worth of work into about 3 minutes, if you will. Among the major things that have happened, and you've heard about this, and you will continue to hear about this, these major steps, is number 1, having scientists around the world poring over those 3 billion ordered G's, A's, T's, and C's to try to understand what is the code embedded in all of those letters. Where are all the functional sequences, and what do they do? I would tell you that 11 years later, we went from knowing this much about how the human genome functions to maybe about this much about how the human genome functions. Eventually, we need to know this much. But in the first 11 years, what we've learned has been eye-opening, inspiring, and motivating for continuing the journey of interpreting and reinterpreting these critical letters that serve as our blueprint. But what was really facilitative in these studies and all the other studies I'm going to tell you about was the fact that we went on a major push to develop better technologies for being able to read out DNA sequence. And here I would point to what has, I think, arguably has been one of the most successful technology development efforts ever undertaken in biology, certainly ever supported by the NIH. And it was an effort basically to develop better methods. Because the first sequence of the human genome was produced in factories such as this, a number of them around the world, at a cost that approached something like $1 billion. But if we were ever going to be able to use this for studying individual people, or certainly eventually in a clinical setting, individual patients, we were going to have far more fancier, more cost-effective methods for sequencing DNA. And the good news is those have come, and they've come in force. And now where it once took a factory to sequence a human genome, it could be done on little devices such as this that plug into little instruments that go on a benchtop or even a desktop. It's been remarkable. And it's not just the cost of sequencing a human genome, it's actually also the speed. So that first human genome sequence, when we generated, it took us 6 to 8 years of sequencing human DNA at a cost of almost $1 billion to complete. That is completely different nowadays, because nowadays I could draw blood from any of you, or even take a little cheek swab, or even take some of your skin cells and isolate DNA from that. And within about 1 to 3 days, at a cost of just a few $1,000s, we could get a complete sequence of your genome. By the end of this calendar year, or certainly early into next year, we expect the time to be down to about a day, and we think within the next year the cost will be down to about $1,000 or less. This is a substantial accomplishment in terms of technology innovation. That has allowed us to do all sorts of things. It certainly has allowed us to understand more about how our lettering differences among our genomes, cataloging genomic variation. When the Genome Project ended, we knew about maybe tens of thousands of spelling differences in people's genomes. we now know about close to 100 million variants that are out there in the population. And it gives scientists then the chance to figure out what those variants are doing and what role they play in health and disease. And these catalogs available on the internet are inspiring research that previously was unapproachable. Much of that research now is being focused on understanding which of those spelling differences in our genomes play a role in disease. Virtually all diseases have important genetic and genomic underpinnings, whether it's rare diseases or more common diseases or diseases like cancer. And we've seen incredible advances in all of those fronts. I'll give you a few examples. For rare diseases, diseases like sickle cell anemia, cystic fibrosis, when the Genome Project began, we knew what gene was broken for 61 of those diseases. Fast forward to today, over 5,000 such diseases we now know the gene that is broken. And we have more to figure out, but that's been remarkable advance. Common diseases, we haven't figured all those out, but in the past 5-7 years in particular, we have developed very important approaches and strategies that allow us to really hone in on specific regions of the human genome, which we now know contain variants, spelling differences, that are conferring risk for all the kinds of diseases that are important and fill hospitals and clinics around the world, like hypertension and Alzheimer's disease, autism, cardiovascular disease, asthma, and so forth. And in diseases like cancer, what you're seeing is going on in cancer research is really breathtaking, because cancer is truly a disease of the genome. What happens in a tumor is derangements in the DNA that make those cells grow out of control. And those same methods we use for sequencing DNA can be applied to sequencing tumor DNA. And as a result, we have seen significant projects that are taking all different types of cancer, analyzing hundreds and hundreds of specimens, making that data available. And now cancer researchers have catalogs of the common derangements that are taking place in each type of cancer. It is changing the face of cancer research, and I predict very quickly will change the face of cancer care. And this has led, of course, to the fifth step, which is probably the earliest one, and you'll see really develop over the next 5, 10, 20 years, is really now using these scientific advances and knowledge to allow us to implement genomic medicine, begin to use knowledge of genomes of individual patients for their clinical care. So just in the last 1 minute, and I could talk about this for substantially longer, but let me just give you just 4 brief highlights, some of which I think you'll hear from others this morning. What are some of the hottest areas? What is the lowest hanging fruit in some ways with use of genomic information for clinical care? I would say the 4 would probably be as follows. First was the thing I was just talking about, cancer. I think already we are seeing for some kinds of cancer, standard of care being genomic testing of that tumor prior to decisions about treatment or predictions about prognosis. I think the number of cancers this will be applied to clinically for standard of care will increase substantially between now and 2020. Another major area of advance we're already seeing is not related to disease, but related to treatment. Pharmacogenomics, a big word, but why is it that different people respond differently to the same medication? It's because each of us have slight differences in our genomes that influence the metabolism of drugs or how we react to medications. And we are beginning to learn increasingly what those spelling differences are and what role they play in how we react to medication. And with a simple genomic test for some medications already, that is now standard of care. Know something about your patient and their unique genomic makeup before deciding what drugs to give them. This is not science fiction, and as a result, I think you'll see increasingly the need to have genomic information about a patient before making decisions what drugs to give them. Third hot area, and you hear about this all the time, whether you watch CNN or New York Times, read the New York Times, or any of the other media outlets, spectacular stories of individuals with devastating rare diseases for which they've gone from physician to physician, medical center to medical center, no diagnosis, unknown what's wrong with them. At that point, for a few $1,000s, sequence their genome, and in a non-trivial fraction of the time, you can figure out what is wrong with that individual. Some cases, it leads to some ideas about treatments. In other cases, it just gives an understanding of what is wrong with that patient and provides an opportunity for scientists to better understand possible mechanisms for that disease based on those mutations in an individual gene. This is now becoming, again, standard of care for situations of individuals with undiagnosed ultra-rare diseases. And I threw in this fourth one, particularly for the topic, the general topic here about women's health, because there are some very exciting things going on in the arena of both prenatal and potentially in the future newborn genomic analysis. Let me emphasize prenatal, because here the technology surge is changing the face of prenatal genomic analyses, where once upon a time amniocentesis or chorionic villus sampling was required to access fetal DNA to do prenatal genomic testing. These powerful new methods using devices such as this for sequencing DNA are now allowing us to detect reliably and analyze fetal DNA floating around in maternal bloodstream. So rather than invasive procedures to access that fetal DNA, a simple blood draw of a pregnant woman is sufficient to be able to do the same testing that otherwise required more invasive methods. This is not science fiction. This is not even 2020. This is already happening now, and many believe it's going to ramp up considerably between now and the end of the decade. So in a whirlwind fashion, that was some of the highlights of what we've done in genomics, what the major steps have been since the end of the Genome Project, and what some of the hottest areas are in genomic medicine. The last slide is really just to tell you one other thing that I think is very relevant for us to think about, is how much genomics has changed in terms of its relevance to different sectors. You know, when I got involved in genomics, which was a little over a quarter century ago, when the Genome Project was just being contemplated, it was really just about biomedical researchers and computational scientists thinking about all this, but it was really just a scientific discipline. I think 11 years ago when the Genome Project ended, it started to shift because healthcare professionals started to see the potential for genomics for clinical practice, and they got engaged and they got involved, and now they're heavily engaged and involved. The difference with now in particular, and I think between now and the end of this decade, is that genomics is going to become relevant to patients and friends and relatives of patients, which means all of us. And all I can tell you is all you watch, at least in the DC area, watch television, some of the commercials, you start hearing the word genomics described when advertisements describe different healthcare networks or different cancer treatment centers. And increasingly, I read about this as well, even in the popular press. Genomics is finding its way broadly across society as a word because it is increasingly going to be relevant to patients for their clinical care, which means it's going to be relevant to all of us. And with that comes important responsibilities to make sure we can communicate and educate the concepts adequately so people can be empowered to understand genomics well enough to have it be relevant to their own clinical care. So I will stop there. I'm delighted to have come here and share this with you, and I look forward to the other speakers. Thank you very much. And we'll bring Rita Caldwell to the podium, and